know. But it is my distinct pleasure and honor to introduce Dr. Nora Volkow as the RTI Fellow Distinguished Lecturer today. And in order to not infringe on our presentation time, I'm not going to give the background that I'd say most of this audience already knows anyhow. She's director of NATA. And I've known Dr. Volkow ever since her very early days at Brookhaven National Laboratory, and had the honor of her choosing one of Mike Kuhar's and my compound to do some of her early imaging studies on. I have followed her studies ever since and have read most of her over 600 publications and 95 book chapters. According to the Web of Science, her papers have been cited by others in their studies over 11,000 times. And I think it's rather striking that Dr. Volkow had 29 publications in 2015 and already has 17 publications thus for this year. And her expertise is not just based on numbers. These publications are in Science, Nature, Proceedings of the National Academy of Science, Neuropsychopharmacology, and Cell. But most importantly, the studies have provided highly useful information about how the human brain works and how it affects and how drugs affect the brain. Dr. Volkow, as you well know, has received numerous awards, and I'm not going to list those here except to point out that she's a member of the Institute of Medicine and the National Academy of Sciences. But saying all this, to me, recently, and I think to many others, the most significant thing that she's done it involves this research and others is the part that she's played at the leadership she had at NATA, and her research is our Congress, Senate, and the President that drug addiction is a brain disease. And as a result, many of our young people now are being treated for their disease, just like ADHD, and not going to jail. In today's presentation, Dr. Volkow will tell us about her research on the project of how drugs enslave our brain.
Good afternoon, everybody. It's a pleasure to be here at RTI. So I thank Dr. Ivy Carroll for having given me the opportunity, alas, at the end, to come and visit this great institution. And I know Ivy for many, many years, and I've always been fascinated by his work, and we've collaborated, and I hope we continue to collaborate. But I did not realize the massiveness of the diverse work that you all do at RTI because you had never invited me. So I actually love being here, and so it is a genuine pleasure, and it was a wonderful surprise. And I hope that I've always, of course, been aware of the scientific impact that you all have. But I look forward to try to actually, by getting to know these other areas of science that at night that we're not so acquainted with, we can establish new opportunities. Because if there's no dialogue, of course, then there's no communication. So it is, I have to thank Ivy for finally inviting me here. So thanks very much. So I'm going to speak today about exactly that, how our brain is enslaved by drugs. But it's actually, we are enslaved by drugs because there is a process in our brain that actually responds to it. And so in studying drugs, we can also get an insight about the neuronal mechanisms that underline some of the behaviors we do, and very importantly, some of the behaviors we do even though we don't want to do them. I mean, that paradoxical situation where we find ourselves having an analytical capacity and make a decision not to do something, and yet we're unable to not do it. So, which can put us into a lot of trouble, and teenagers are particularly prone to getting into trouble. But I think that it does give us an insight. And so what I am going to be speaking today about drug abuse and addiction pertains to something that is very fundamental on how our brain works. Now, it so happens that drugs actually are affecting one of the systems that is most relevant in regulating our behaviors, which is the dopaminergic system. In the past, we've actually just had this very simplistic notion that dopamine is equal to pleasure. So if I have pleasure, I will do the thing. If I have displeasure, I will not do it. And while there is truth to it, it's also come to understand that what dopamine is signaling goes beyond just pleasure. What dopamine is signaling is actually saliency, something that is important, something that is relevant for that particular organism or creature, that in a way that is relevant for survival. So in long and behold, of course, food is signaling a reward, a reinforcement, and that motivates you. So when you activate the dopamine system, what you are doing is setting into action the drive that will make you do behaviors to actually ultimately be able to consume or do whatever is associated with pleasure. But as I say, it's beyond pleasure. So if something is actually novel that you don't expect, dopamine is stimulated, and that allows you to engage your attentional mechanism toward it and give it priority. When something is very aversive, that also engages the dopaminergic system. And what it does, it enables you to learn very, very rapidly that that's aversive and can have untoward consequences. And that learning actually requires one exposure. You will not do it again. So dopamine has the capacity to engage multiple circuits in the brain that will motivate your actions, that will lead you to learn those associations in such a way that you can recognize them as important or not in order to approach them or avoid them in ways that are very, very fast and efficient, that we call conditioning. But it also allows you to maximize the utilization of your neuronal resources to things that are relevant. So if something is very predictable and you know it's going to be there, you don't want to engage your attentional system because it doesn't really matter. On the other hand, if it's novel, you should, because you don't know if it's going to be helpful or not, or harmful. Now, drugs activate these dopaminergic pathways, and they do it in ways that are much more potent than any of the natural reinforcers or even stimuli that actually are activating this dopaminergic system. And that's what's so dangerous about drugs. And all of the drugs that have the capacity to produce this supraphysiological stimulation of that dopaminergic system, predominantly of that dopaminergic system, the one that targets the nucleus accumbens, which is a hub in the network of reward and reinforcement and saliency. That system, all of the drugs that can do that at the supraphysiological level have the capacity to produce addiction. And so that is, I think, from the basic literature, something that provided us a really important insight about why what may be happening in the brain of a person that's addicted. And there you have some of the first microdialysis experiments showing that food increases dopamine. Of course, it's rewarding, necessary for survival. It's and in the extent at which increases dopamine, the animal is going to engage on behaviors on getting it. But look and compare with amphetamine. It's actually amphetamine is much more potent. The effects are much longer lasting. And this is what I was calling before supraphysiological, can then trigger adaptations that in those that are vulnerable can result in addiction. The big question mark has been, what are those adaptations? Why is it that if any one of you were to take a drug, and you can call it alcohol or you can call it whatever you want to, it will increase dopamine, it will activate these nucleus accumbens, but it's only in a person that's addicted will it generate a desire of more and more and more. It's almost like insatiable craving. So what is the difference? If in both an addicted and a non-addicted person, you're increasing dopamine, why is it that in one you're generating that insatiable craving and in the other one it's not? And so I'm going to give you some insights in terms of what we've been able to uncover by using brain imaging, in particular positron emission tomography, to try to uncover what it is. To start with, everybody has been very interested on the notion, yes, all of the drugs that increase dopamine in the nucleus accumbens, striatal regions, can produce addiction. Okay, does that happen in the human brain? And using imaging, which is actually was allow us to test this in humans, because you cannot put a little electrode and start to actually do microdialysis or electrophysiological studies in the nucleus accumbens. That's invasive in a human. But you can do imaging, and it's actually quite straightforward. The way that you measure changes in dopamine is indirectly by looking at the occupation of the dopamine receptors. So you use a ligand that is labeled with a positron emitter, carbon 11. The one that we've used, the one that's most extensively used, is raclopride. Raclopride is an antagonist that binds at the dopamine D2 and the dopamine D3 receptors. It's relatively low affinity, so it can only bind to those dopamine receptors that are not occupied by dopamine. Since normally, when you're not taking a drug, 85 to 90% of those dopamine D2 receptors are empty, you can actually then measure the difference in the binding of your radioligand when you give a placebo and you have 85 to 90% of those receptors free and raclopride binds happily, versus when you give them a drug. In this case, the drug that we've been extensively studying is methylphenidate, and we've actually, in this case, started by using intravenous methylphenidate because if you look at the pharmacology, methylphenidate is probably the most similar drug that you can get to cocaine. And I was very intrigued by that because I says, well, if cocaine is believed to be addictive because it blocks the dopamine transporter, which is the mechanism by which dopamine is recycled back into the dopamine terminal and terminates its action. So if you block it, it cannot be recycled, dopamine accumulates, that stimulates, and that's what traditionally everybody was saying, that's why cocaine is rewarding. So I was saying to my brain, well, if methylphenidate can do that as well as cocaine, what is it that we're not understanding? Or are we giving methylphenidate, which is Ritalin, to children, and which is a drug like cocaine, or there's something else that we don't really know? So that's why we started. I actually got very obsessed with methylphenidate. Methylphenidate blocks the transporter, dopamine accumulates, and then it binds to these receptors. So when you give your raclopride, binding goes down. So then you compare placebo versus IV methylphenidate, and you can see that decrease in the binding of your raclopride because these are occupied with dopamine, so it cannot bind. It's like a seat is occupied by someone, you cannot sit there. And after appropriate mathematical modeling, you actually can quantify relative changes in dopamine. And lo and behold, when we said, okay, well, all of these studies in animals, in rats, in monkeys have shown that the increases in dopamine are necessary for the rewarding effects. What about humans? So we did one of the first studies to evaluate this, and we used intravenous methylphenidate, which, by the way, cocaine abusers don't distinguish from intravenous cocaine. You can ask me questions about it, but in animal models, it's basically the same. They don't discriminate intravenous methylphenidate from intravenous cocaine. We injected it. Intravenous methylphenidate gives you a very strong high. And cocaine abusers don't, as I say, don't differentiate it from intravenous cocaine. And then 1 minute later, we injected C11 raclopride. And we measured changes in dopamine produced by intravenous methylphenidate. And this is the relative changes in dopamine, as assessed with with raclopride by the changes in specific binding versus the subjective experience of high. And we demonstrated that indeed in individuals in whom intravenous methylphenidate produced a robust change in relative concentrations of dopamine, you saw a very intense high. In those that it didn't produce any subjective effects, indicating that in humans, just like in animals, the ability of this drug, a stimulant drug, to increase dopamine was directly associated with the intensity of the high. Similar findings actually had been published a couple of months before by the group at Yale University using intravenous amphetamine, where they also showed that the magnitude of the dopamine increases were associated with a high. And since then, other investigators have actually shown that other drugs like alcohol and nicotine, the magnitude of the dopamine increases is related to the reward. However, I have been, nonetheless, when a thought or a question comes into my brain, it doesn't let me go. And I was very intrigued about the notion about here is methylphenidate is the most widely prescribed psychoactive medications for children in the United States, methylphenidate, Ritalin. And I said, what is it that we are not actually really understanding? And people were saying, well, when you're giving methylphenidate, you don't inject it. That's correct, and that's very important, and I'll mention why that's crucial, in fact. But their belief was actually that when the hypothesis was when you give oral methylphenidate, it's rapidly metabolized into Ritalinic acid. And Ritalinic acid, the belief was, has low affinity for the dopamine transporter. So it's a very poor stimulant medication. And so the hypothesis was that methylphenidate, when given orally, actually was not increasing dopamine. In fact, there were hypotheses that they were saying it was decreasing dopamine, and that's why it's therapeutics for ADHD. There were many people that believe that stimulant medications, methylphenidate oral, was actually decreasing dopamine signaling. So we decided to do the study. So, okay, if this is intravenous, what happens when we give it orally? So we gave oral methylphenidate to adults. These are studies in adults, but we had to wait 60 minutes because if you give the drug orally, it takes 60 to 120 minutes to peak in the brain. And then we measure changes in dopamine. And to our surprise, we show that oral methylphenidate produced a significant increase in dopamine. So even though it was being metabolized with urichalinic acid, you still show a very robust response. And in fact, some of the individuals had changes in dopamine that were indistinguishable from those that you were observing with intravenous methylphenidate. The difference was that with oral methylphenidate, you were not producing a high. And you can see that here. But the fact that you were not producing a high was not because you were not increasing dopamine, but because actually you were increasing it. But so the question is, why were you not producing a high? And that is where, again, at the essence of, I think, an understanding why drugs are rewarding. What is it that makes a drug rewarding? We had that traditional simplistic perspective that drugs are rewarding because they increase dopamine. Well, if it was so simple, then these oral methylphenidate had produced a high, and it didn't. And these were the issue of the route of administration jumps in immediately. And we knew already from work from Schuster and that the rate at which drugs get into the brain determines their reinforcing effects. In other words, the faster the drug gets into the brain, the more powerful its rewarding effects and the more addictive it is. And that's why the route of administration is so important in terms of addiction. If you take a drug injected or smoke, the drug gets into the brain incredibly rapidly, and those are the most addictive routes of administration, the most dangerous. On the other hand, if you take it orally, it takes its time until it reaches the brain. Plus, there are differences too of bioavailability. But Schuster had done studies where he actually controlled for bioavailability differences and clearly had shown that the rate of entry profoundly affects the rewarding effects of drugs. And indeed, we now know that is the case. And here you have just data showing the pharmacokinetics of C11 methylphenidate, which we had labeled with carbon 11, substitute one carbon for the other, injected it intravenously versus orally. And you can clearly see the differences in the rate of uptake into the brain. And that rate of entry into the brain, which translates into rate at which you are changing dopamine, we had shown by other experiments that are not presented here, is basically what is associated with the high. The high is associated with very fast changes in dopamine. If you don't produce those fast changes in dopamine in the brain, you're not going to get a reward response. And I was for many years struggling in my brain, trying to understand why. And my brain was becoming very convoluted. And I was actually saying, and I got this fixation because I actually, to me, it's very striking that you pay attention to the refrigerator noise when it stops making it. And I started to read about it, and there are cells effectively in the somatosensory cortex, in the auditory cortex, because this is also true for somatic sensations. You may sometimes feel the sensation when it's no longer there, that respond to a discontinuity in the stimulus. So in my brain, I was sort of saying there must be something like that reward is associated with discontinuity. And I was trying to figure it out. I never figured. And one day I said, oh my God, come on, Nora. It's very obvious of what it is. And I was so upset because it took me a while to understand, and it was very obvious. And I use this as a teaching lesson for when I speak with postdocs. Look for biology for your answers because they are in front of you. Just look at them. Sometimes we don't see things. We just they are transparent, but they are there. Dopamine cells, okay, we know that dopamine is important, so let's look at the dopamine cells. Dopamine cells have two main modes of firing, just in a simplified way. The tonic dopamine firing, which is a spontaneous tonic dopamine cell firing, that actually increases dopamine when the cell fires. There is release of dopamine, but the release of dopamine is relatively modest. And that's one. And that tonic dopamine cell firing, which is actually between 2 and 10 hertz frequency, allows us to be receptive to stimuli. So right now, and to sustain attention and to sustain effort. So right now, my ability to engage you depends very much on my capacity to keep that tonic dopamine cells working properly. If you are very tired and sleepy, then the dopamine cells may go slower, and then I may not get your attention. Or if I sort of basically get to speak about something that you're not interested, you'll start to mind wander. Your dopamine cells will disengage. But if something gets very exciting and very, very striking or novel, then the dopamine cell starts to fire very, very fast, very short trains, milliseconds, but they go from 2 to 10 to, now we know, 15 to 50 hertz. And that produces a very sharp, very high increase in dopamine that's very short lasting, but very high. Now, what that does is actually associated with saliency, where it's reward, aversion, novel. And what it is signaling, that allows you to signaling a different mechanism. Because we have been very, again, sometimes, I mean, we get an hypothesis that dopamine increases, and that's why it's rewarding. But dopamine signals throughout 5 different types of receptors, though they have any importance. And again, let's look at biology. Well, most of our research has been on dopamine D1 and dopamine D2 receptors. Dopamine D3, we know, are very important for drug reward, but we've studied them less, but they are very, very interesting. Dopamine 4 and dopamine 5, much less investigated. The concentrations are lower, but we have a lot of data on dopamine D1 and D2. What do we know? Dopamine D2 receptors are very high affinity receptors. So you stimulate them with relatively low levels of dopamine. Ergo, you stimulate them when dopamine cells are firing tonically. So in order to sustain your attention, dopamine, even if it's not very high, is going to stimulate those dopamine D2 receptors. In contrast, D1 receptors are low affinity. So in order to stimulate that, you need this spiking of dopamine. And when it spikes, the signaling then can generate a different type of processes than those that are linked with dopamine D2 receptor signaling. In particular, reward, in particular, conditioning. So in order for a stimuli that increases dopamine to produce a conditioning, which is this learning process that allows you to associate a stimuli with a reward or a stimuli with an aversive response, that conditioning, which, as I say, can happen on one-time exposure. That's D1 receptor mediated. So in other words, for drugs to be addictive, they have to emulate phasic dopamine cell firing. They have to stimulate dopamine D1 receptors. So then the notion of just increasing dopamine is not sufficient. It has to increase it in ways that it's activating the dopamine D1 receptor system. And I'll come back later. The dopamine D3 receptor system is also very, very relevant. And they actually, the D1 and the D3 receptors are expressed in the same cell, and they facilitate actually signaling one from the other. And that's why the D3 receptor is important. You knock down the D1 receptor, create a D1 receptor knockout mice, you profoundly affect the rewarding effects of drugs. You knock out the D2 receptor, knockout mice continue to self-administer drugs. So we now know that the D1 receptor is indispensable stimulation for rewarding effects of drugs, whereas the D2 receptors actually are not indispensable. An optimal reward actually happens when you're stimulating both of those systems. So this is very, very, very important because it gives us also an insight in terms of the adaptations. And this is an optical imaging where we can actually, by using fluorescence, separately identify cells in the nucleus accumbens that express the D1 receptors versus cells that express the D2 receptors. We can give those animals cocaine, and by measuring calcium through a fluorescent probe, we can determine how a drug differently affects these two D1 versus D2 receptor cells, which actually, again, are signaling through different pathways. The D1 is what we call the direct striatocortical. The D2 is the indirect striatocortical. You give cocaine, D1 cells actually are stimulated when you give them cocaine, and dopamine goes up. It does a very sharp increase, and then it sort of plateaus. D1 inhibits the D2 when it's stimulated by dopamine, in this case by giving cocaine. It actually inhibits neuronal firing. and the effect is much lower. And this actually, again, highlights what dopamine is doing in this reward circuit. It's stimulating very rapidly the D1, whereas it's actually deactivating the D2 receptor indirect pathway through a much slower process. So in this line of thinking, then, how does that relate? How do we take this understanding about what drugs, why drugs are rewarding? They increase dopamine, but they stimulate the D1 system. How about addiction? And for many, many years in the field, it was believed that actually, and still many believe, because it takes a long time for people to actually accept findings. I mean, I think that some theories are so appealing that no matter how the data looks, you want to ignore it. And so it was very, very nice to say a person is addicted because in them, the rewarding effects of drugs are more potent. In them, that's the theory, the drugs of abuse produce much larger increases in dopamine. And therefore, they can actually enjoy them more, and that's why they take them. And again, they enjoy them more, and then that's why they take them. But then again, it's a very myopic perspective, because if you've ever been with a person that's addicted, you've come to realize that in these individuals, they're taking the drugs not to get high or feel great, but actually to feel normal, because when they're not taking the drugs, they feel miserable. So this notion of taking a drug initially, because it's rewarding, to a state when you become addicted, when that drug is no longer rewarding, but you actually are making it to feel not as miserable, is the transition that actually has led, that still has not permeated in the public or in the healthcare system that would allow them to recognize that a person that's addicted is not taking the drug because they are choosing. They're taking the drugs because they actually need them and their brain is processing them. So what goes on? So in terms of the hypothesis, I love the hypothesis that addiction is because you are addicted to the drug because it's more rewarding and there's going to be more dopamine when you take it. And I sort of say, beautiful hypothesis. And if you're writing grants, it's wonderful when you have a beautiful hypothesis because you'll convince everybody resonates. I say, oh, wow, yes, it's great. And I did put the grants sort of to postulate that cocaine abusers would have increases in dopamine when they took the drug. And I got very, very high score. I got funded. And these are the results. This is a normal control. And this is, as you see, an old paper. And we showed normal controls. And then I look at the cocaine abusers, and I was shocked of my brain. Because you can see the normal with placebo methylphenidate, you see that decreases. But in these cocaine abusers, you see no difference. And this was antithetical to what everybody else was saying. When you quantify the percent change in the specific binding of raclopride, you see that the response of the cocaine abusers is half that of the controls. And this was very shocking, and the community did not like it because it was really antithetical. We were showing not only that there was not an enhancement of dopamine signal in people that were addicted, but that they were actually significantly attenuated. And then the group at Columbia went on to replicate this, and we've replicated this in alcoholic subjects. But one of the criticisms that actually had been voiced very early on, and I mean, it's definitively an important one, was they said, well, Nora, how do you know that the reason why you were getting these attenuated responses is not just the fact that this is happening because they are addicted, but because you study them during the withdrawal, and that this reflects withdrawal? And a very, very valid question. So recently we went on to do the same experiment, but in active cocaine abusers to address it. And so here is obviously a much more recent paper. And what you're seeing is a better scanner, and the images have been already normalized for non-specific binding, for metabolism of the radiotracer, and they are average. So this is one subject, these are average. So this is the placebo, the average images for our 17 controls in placebo, 17 controls on methylphenidate, and this is the cocaine abusers. And it's a much better way of looking at it because you then never know if you selected your best subject or your worst subject. This is not selection sensitive. It's just the average. You see the placebo and the decreases in the binding, and this you actually see the cocaine abusers. This is the control group, and it's very difficult to visually see any difference. And when you quantify, these are active cocaine abusers, you can see that, in the ventral striatum, where the nucleus accumbens is located, the response of the abusers actually is 20% really close to 20% that of the controls. It's profoundly attenuated. So we presented this data, and someone in the audience says, well, Nora, how do you know that you are not getting these responses because you are actually getting the subjects in the PET scanner environment, laboratory environment? And if you've been into a PET scanning environment, you'll realize that this is a very valid question. It's not very enticing to get you excited about getting a drug because you are lying there with catheters all over the place and everybody's in white. So, okay. So yes, well, let's test this hypothesis. So we carried on another study in active cocaine abusers, but instead of just giving them IV methylphenidate by itself, we gave them IV methylphenidate into two different days, two different conditions. On one of them, we exposed them to cocaine video that was tailored to the way that they administer cocaine, to their ethnicity, and to their route of administration. And we had shown by pre-testing this particular video that it produced significant craving, and actually by itself, it was able to increase dopamine. And then on another day, we gave them the IV methylphenidate, but we gave them a video of nature scenes. And we asked the question. The hypothesis was that when you engage them into getting into the mindset of seeing others take cocaine, which creates craving and expecting to get high because you are seeing the visuals, that we would be able to see an increase in dopamine that was actually significantly larger than we had been able to see before. So we did a study, and this is the results from this study. I'm showing them with a different, using statistical parametric mapping, which actually identifies on the upper row in different levels of the MRI from top to lower parts, the areas of the brain that the raclopride binding was changed by intravenous methylphenidate in healthy control subjects. And so you see different planes, and the nucleus accumbens you can get located between these two planes. And you can see a very robust change in raclopride binding because dopamine is going up. And the significance levels here, so it's a statistical map at 0.001, and this is your percentage of changes. When I do the same threshold of significance for the cocaine abusers, whether I use a Q or a neutral, I don't see anything. So against all of the statistical gurus, I brought it down to 0.05, and that's very, very bad. But I didn't see anything, so I said, okay, I don't see anything. Let me bring it down to 0.05. And then I start to see these little spots in the ventral striatum. Normally, I would not even dare to present that at a 0.05 level. But why I'm presenting it, and I think it's very, very real, is that when subjects were real, that even though it's profoundly in the ventral striatum, and it didn't matter where we had Qs or non-Qs, indicating that the blunting of the ability of intravenous methylphenidate to increase dopamine in the striatum and nucleus accumbens is not due to withdrawal. Number one, it's actually even more severe in active cocaine abusers, and is not due to the lack of Qs. That it represents a true hypodopaminergic state when they are actually consuming the drug. And then you'll say, wait a second. I mean, if you are showing that, and the data shows it's not just us, others have shown that increasing dopamine in animals or humans is indispensable for reward, what the hell would a cocaine abuser even bother if the signal is so very, very low? Well, look at this. The issue that's interesting, and this is the ventral striatal slice, the delta on the controls, the areas where it's changing dopamine, these little points in the ventral striatum in the cocaine abusers. And you see what's very, very interesting is that in them, you see that the larger the changes in dopamine here, which is a decrease in the binding, the larger the decrease in the binding, which means the largest the increases in dopamine, the more intense the craving. And this is actually paradoxical because they are very attenuated. That's what differentiates them from controls. And yet, the greater the increases in dopamine in the ventral striatum, the more intense the craving. And how does one interpret that? I mean, it basically tells you that even though it's very attenuated, it's still able to increase dopamine. And in so doing, it's triggering the craving. So what could it be? Well, there are two things that I think could it be. One of them is with raclopride, we have a terrible disadvantage because you are averaging changes over 30 minutes. And yet I told you that the high is produced that very intense moment of phasic dopamine self-firing. We're not measuring D1 receptors, we're measuring D2 receptors over 30 minutes. So could it be possible that, and that's one of the possibilities, that during the first few seconds, there is a sharp increase in dopamine that then maintains the high to a certain extent and produces the craving. But the other one that also, and is not per se exclusionary from that hypothesis, is raclopride binds to both D2 and D3 receptors. And in the nucleus accumbens, the concentrations of D3 receptors are very, very high. In the caudate and the putamen, they are very few, so you can dismiss it. Raclopride is predominantly D2. But in the nucleus accumbens, D3 receptors account for at least 50% of the binding of raclopride. And we know from animal models that D3 receptors are overexpressed in animal models of addiction. So we believe, actually, that what is accounting for this association is that these changes in dopamine that we're seeing is driven by D3 receptor signaling. D3 receptor signaling interacts with D1 receptor, facilitating the transmission of those rewarding signals. So that's what we believe is going on. But also highlighting the relevance that D2 receptors by itself appear to have a very profound decrease in signaling when an individual is taking the drug. And we've actually gone on using optical imaging where you can actually manipulate all of these parameters and look at the D1 receptor expressing cells that form the direct striatocortical and the D2 receptor cells that form the indirect striatocortical, and differentially observe how these systems adapt to chronic administration of cocaine. And we have shown, actually, and others have shown too, this is our data, that again, using a calcium imaging, D1 cells, when you give cocaine, increase. And when you give chronic cocaine and then you give them acute cocaine and intoxicate those animals, you see an attenuation of the increase, but you still see the signaling. In D2 receptors, you see the decrease in calcium signaling with an acute, and when you give chronic cocaine to an animal, you basically completely abolish the D2 receptor signaling. So while we are observing, and others have observed, attenuation of D1 receptor signaling with chronic cocaine, the attenuation on the D2 receptor signaling is actually much more accentuated. And what is transmitted in the striatum, in nucleus accumbens, or in any other parts of the striatum in terms of dopamine signaling, is stimulation of these two pathways and a balancing on one another. And the idea right now in the addiction field is that it is the D1 receptor system that accounts for the rewarding effects of drugs, and the D2 receptor system is a system that opposes them. And so when you are in a chronic state, when you are in an acute state, you take cocaine, the D1 has, this is D1 over D2. You have an initial period where D1 is dominating, but with time, the D2 receptor goes, actually activity continues to go down. So you basically create a baseline state. And we believe that this terminates the desire to take more drug. When you are in a chronic state, D1 or D2 receptors continue to go up, and there is a predominance throughout the 30-minute period that we were doing of the D1 receptor signaling over that of D2. And we believe that this may be one of the mechanisms by which you actually do not satiate. You actually are continuously stimulating the D1 system with no opposition from the D2 receptor pathway. Now, that's the reward system. What about when you are, this is when someone is intoxicated. But when you are addicted to drugs, you're not always intoxicated. And in fact, one of the big challenges, and this is important because that's when you can do a therapeutic intervention. So what goes on in their brains when they are not intoxicated? We better know, because that's when you're doing the therapeutic intervention, and that's when you can actually help them, prevent them from relapse. So we've shown that there is this attenuation, profound attenuation of dopamine signaling through the D2 receptor pathway, certainly, when you are intoxicated and you're addicted. But what happens when you are not there under the effects of the drug? well, what has happened to the dopamine system? So the dopamine system is active whether we're taking drugs or not, and whether you're an addict or not. So we were very interested on actually understanding how is the dopamine D2 receptor system working when you're not intoxicated, and what are its consequences on brain function. So in order to measure brain function, we have used as a marker brain glucose metabolism. In animals, we use calcium indicator. In humans, we use brain glucose metabolism because it's a very sensitive indication of function and activity. And we've actually studied, and Trice is a very interesting that I have this slide here, but in any case, that's a corruption of the program. What the concept has been that when you take a drug, you actually are overstimulating dopamine, and the system tries to compensate itself. And we know from studies done in animals that if you give a neuroleptic, which blocks these dopamine receptors, you compensate by creating more. But if you give stimulant drugs in animal models, you compensate by downregulating dopamine D2 receptors. So we wanted to know if, and again, when a person is not intoxicated, whether these D2 receptors are influenced in a different way, are affected in cocaine abusers or drug-addicted people. We've been studying them for many, many years, at least 25 years, and we have consistently shown that what you are observing here. Others have shown what you are observing here. Animal models, whether it is rats, mouse, or non-human primates, have shown this. In general, cocaine abusers, it's interesting, it's corrupting it here again. Methamphetamine abusers, alcoholics, or heroin abusers, cigarette smokers, nicotine, you see a downregulation of dopamine D2 receptors. So this is control, this is addictive, and this is data sets from 2 groups of cocaine abusers tested during at least 3 weeks after detoxification. So this is quantified as dopamine D2 receptor availability in controls, in pink, in cocaine abusers in green, using different PET radio tracers, and plotted the availability of D2 receptors as a function of age. This is N-methylspiropyridol. This is fluorinating N-methylspiropyridol. This is C11 raclopride. You see that as we grow older, D2 receptors go down, whether you're a cocaine abuser or a control, here or here. These were studies in patients, inpatients. These were outpatients on treatment to ensure that they were not taking drugs. And you can see that cocaine abusers have lower levels of dopamine D2 receptors. And that was a very interesting finding. And it was one of the first findings that actually led to the conceptualization that addiction is a disease of the brain, that you can actually characterize biochemical changes in the brain of people that are addicted. What was very interesting in my brain, of course, is you show a finding like this one, and you say, if it's so consistent, and it's showing that there's decreases in dopamine D2 receptors, can you say that that's the addicted phenotype? And I would love to say, yes, that's the addicted phenotype. It would be fantastic, right? But it's not. And it's not because you can actually see that there are addicted people that have low normal levels, and there are normal people having lower levels. But overall, it's a very consistent finding. But it's not that you categorically differentiate one from the other. So when you have data like this, what is it that it tells you? I mean, what it tells my brain is that having low levels of dopamine D2 receptors is making you vulnerable to a phenotype, making you vulnerable to a phenotype on addiction, which is compulsive use of drugs, inability to stop taking them. That's what addiction is about. And or alternative, that having high levels of receptors may be protecting you. So how do you test that hypothesis? This is in imaging. You actually are basically just making a description. The way that you could mechanistically test this in these people, if I can increase the levels of dopamine D2 receptors and affect the phenotype of self-administration of drugs, then I know that they are causally linked. I cannot do that in humans, but we've done it in actually rats. And here you have 2 different studies that we've done in rats for alcohol. We've done the same experiments with cocaine, and the results are the same. The results are basically the same. They vary in terms of the magnitude and the duration, depending on the strain of rats that you're working on. So this is data for Sprague dollies. This is data from alcohol-preferring rats. And so I'll explain the protocol here. With a Sprague dolly, we make these animals addicted to either alcohol or cocaine. When they are self-administering high doses of alcohol or cocaine, We then inject them with an adenovirus inside of which there's the D2 receptor gene, stereotactically into the nucleus accumbens. Because we have imaging, we can actually measure how that changes the levels of dopamine D2 receptors. So I was saying, if I could increase the levels of dopamine D2 receptors in addicted people and change the phenotype, then I know that they're causally linked. Well, I can do this with this gene therapy. Dopamine D2 receptors are increased by 50% within 4 days of administration. And then, unfortunately, with time, they go back to baseline. And it has to, this is limited because we're using an adenovirus. We don't have another vector. But in the question then is, when you are able to increase the D2 receptors, what happens to their drinking? And this is percent change in alcohol intake from 0. So this is negative, and we see a profound reduction in drinking. And as the receptors go back to baseline, the drinking goes back to baseline. You don't see an effect if you only inject the adenovirus with no gene inside. So we needed to control for non-specific inflammatory responses. We don't see anything like that. We see the same phenomena with a DT overexpression in the nucleus accumbens. In animals, the alcohol-preferring rats have been bred for the preference of alcohol. They love alcohol. And in them, even in them, and they've looked at the genetics, and the D2 receptor is not involved in that one specifically. The gene for the D2 receptor is not involved. But in these animals, if you overexpress the D2 receptors, you significantly change the amount of alcohol drinking in these animals, indicating that overexpression of dopamine D2 receptors is protecting these animals from the consumption of high quantities, high doses of alcohol. As I said, we've shown exactly the same thing, or cocaine. There's something about having low levels of dopamine D2 receptors that leads you into this pattern of consumption of high doses of drugs. We've shown it for alcohol and cocaine, and a group of Trevor Robbins and Everett in Cambridge have shown that if you select animals on the basis of the levels of dopamine D2 receptors, you can predict which animals are going to be administering compulsively cocaine, those that have low levels of dopamine D2 receptors in their ventral striatum. So having low levels of dopamine D2 receptors makes you vulnerable for compulsive administration of these drugs. Why? Right? I mean, it sounds very nice, dopamine D2 receptors, but how can one protein have such a profound effect on a behavior that is crucial for addiction? Well, we've been working with in imaging, and again, look at biology and try to figure out and get your responses. The dopamine D2 receptor indirect pathway has been extensively studied by researchers that are involved in Parkinson's disease. Parkinson's disease actually damages the dopamine cells, and that has led them to really, they were the ones that delineated these very elegant direct and indirect pathways. And we know that. So the D2 receptor is indirect. You have the striatum. Dopamine D2 receptors sit in GABA cells, which are inhibitory. And when dopamine stimulates D2 receptors to GABA cells, it inhibits them. And when you inhibit an inhibitory cell, that it disinhibits it. That's normal. But in people that are addicted, there are low levels of dopamine D2 receptors. So dopamine cannot disinhibit this pathway that goes from the striatum to the globus pallidus external. So there is an increased inhibition because you have low levels of receptors. You have an increased inhibition of this inhibitory pathway. That means it disinhibits it. And this pathway goes into the subthalamic nucleus, which is stimulatory. Since it's disinhibiting it, there is increased stimulation of the subthalamic nuclei. Subthalamic nucleus projects into the substantia nigra reticulata or the globus pallidus internal, which is inhibitory. So you stimulate an inhibitory pathway, you enhance inhibition. And this is the pathway that goes into the thalamus. So there is enhanced inhibition for the substantia nigra reticulata into the thalamus. Thalamus is stimulatory to the cortex, but there is enhanced inhibition. That means decreased excitation. So you could predict that when you have low levels of dopamine D2 receptor under baseline resting state, when you're not intoxicated, your frontal cortex is going to be hypoactive. And indeed, when we've done studies in people addicted to a wide variety of drugs, when they are not under the state of intoxication or we're not exposing them to cues, we see a profound decrease in brain glucose metabolism. You can see it here, and you can see it here. Moreover, we have consistently shown and replicated, and again, there's a contamination of the slides here, but it actually shows exactly the same. The lower the levels of dopamine D2 receptor, the lower the activity in prefrontal cortical regions. And we've seen this in cocaine abusers, in 2 independent groups of cocaine abusers. We've seen it in methamphetamine abusers. We've seen it in alcoholics. The lower the levels of dopamine D2 receptors, the lower the activity in prefrontal areas of the brain, as would have been predicted by that understanding of the indirect pathway. And this is actually a study that we did, not in people that are addicted, but in individuals who have a family history of addiction. And we did exactly the same thing. We measured dopamine D2 receptors in the striatum, and we measured brain glucose metabolism. And again, you see the areas of the brain using statistical parametric mapping and showing it there, where the dopamine D2 receptors were associated with activity in the brain. Overall, in the yellow area, that's where it is significant. You can see the medial orbital frontal cortex, the lateral orbital frontal cortex. That's where the correlations are the strongest, and along with the anterior single-edge arrows. The higher the levels of dopamine D2 receptor, the greater the metabolic activity in these brain regions. When you look at the whole brain, we were also able to identify that there is an association with the insula, and clearly there is an association with the dorsolateral prefrontal cortex. So in an individual that is addicted to drugs, the decreases in dopamine D2 receptors are actually associated with reductions in activity of prefrontal areas of the brain that are necessary to exert self-control, that are necessary to regulate desires, to regulate emotions. This is the pathway that allows us to inhibit our amygdala when it gets out of control. And the prefrontal cortex can actually rein it in. But if that system, if the cortical areas are not functioning properly, you're not going to be able to regulate and modulate those reactions. Interestingly, in this group of individuals that have a family history that were not alcoholics, the levels of dopamine D2 receptors were not decreased. And that allowed them to have perfectly normal levels of glucose metabolism in this prefrontal cortex. I hypothesize, it's purely speculative at this time, that these individuals were to drink alcohol, that would result in a downregulation of dopamine D2 receptors and a reduced activity of prefrontal areas that then would make them very vulnerable for the compulsive administration of alcohol. So this has led us to this model. This is actually a simple model that takes into account this very simple system that we all deal with on a daily basis that is crucial and fundamental for survival. A subcortical system that drives our emotional regulation, which is very relevant, is very primitive. It allows you to fight, to fly, to actually to take risk when actually it may mean that we may find food or not. And that it's driven in part by driving the motivation to do things. When we get exposed to something we like, we've been conditioned to it. The conditioning system, which involves amygdala, activates the nucleus accumbens. But we also have our prefrontal cortex that allows us to analyze the situation and make a decision. I want this, but maybe I shouldn't do it because then I will feel guilty, or it's not socially appropriate, or it's politically incorrect. And I find myself many times saying, Nora, it's politically incorrect, don't say that. I'm pretty good at not saying things that I shouldn't be saying. So my prefrontal cortex can overcome an emotional reaction that I said, it's time that someone stands up and says it. It's, wait a second, Nora, wait a second. And I don't. I mean, so it's as ubiquitous as that. It's not just about rewarding things that we traditionally associate, but the way we engage in multiple things, something that generates an intense, passionate reaction that drives your behavior, actually is engaging this system. When you're addicted, unfortunately, your prefrontal cortex is not functioning very well at all. It's hypofunctional. And you have been hypersensitized to conditioned stimuli. So I reported to you that there is a degradation of the ability of drugs to increase dopamine when you're intoxicated overall. But there is an enhanced sensitization of the conditioning response. And we now know and understand something that it's still hard to really swallow, that the main driving in a lot of the behaviors is these conditioned responses. And it's not just in addiction, it's actually in food. When you see a chocolate, and I like to say, oh, I want to eat the chocolate because of the chocolate itself. The reality is I want that chocolate because I become conditioned to it, and my whole body is expecting to get that. And so what's driving me, and we all see it, because many times you are not thinking about eating a chocolate, but someone shows it to you and you immediately want it. You've actually triggered that condition. Or you go to a place where you had that chocolate before and you immediately want it. Or that certain time of the day when you had the idea of taking a chocolate, the next day you want the chocolate at that time. You're conditioning. That drives our behavior. That's sensitized in the person that's addicted. So even though the individual may cognitively be aware that they don't want to take any drugs anymore, their ability to actually control and regulate that process through this mechanism that inhibits amygdala, hippocampus, and regulate release of dopamine in the nucleus accumbens is impaired. And that's ultimately why the person that's addicted, even though they don't want to take it, is unable to stop it because the prefrontal cortex is actually non-functioning. And it's almost like I like to think about it in terms of the pathways that we know with fear. You're walking in the forest, so actually your heart starts to go ballistic on you. You don't know why it's ballistic, and you see there's a branch, a black branch over there. And it takes you a while to see that it's a branch. By then, your heart has already reacted, thinking it is a snake. So what happened is that the stimuli, which is conditioned, activated the amygdala before it activates the cortex. The amygdala sends signaling through the sympathetic pathway to activate you in order if you have to fight or if you have to go to flight before you are conscious that you are doing it. The consciousness comes later. In a person that's addicted, the process of conditioning have generated that response, which explains why many patients will tell you, Doc, I did not want to take the drug. They sort of been in jail, they are released, and says, without even realizing it, I already had the drug in my mouth. The inability to stop that behavior is because it has been short circuit out of the consciousness, generating an automatic response. And this understanding, of course, gives you an insight of therapeutic interventions, which obviously are targeting not just one system. It's not just that the reward system is completely disheveled and by the use of drugs, making you much less sensitive to the actual consumption, but triggering an enhanced sensitivity to the conditioned responses. There is also this profound disruption of the prefrontal executive control networks in the brain that allow you to regulate yourself. And as George Cook has eloquently shown, there is also an enhanced function of the stress circuits that makes you very, very vulnerable to negative stimuli that then can trigger a relapse. So interventions that actually aim at these different elements are actually likely to, by helping counterbalance the abnormalities produced by drugs, can help a person be able to control some of these very automatic responses. And with that, I want to thank you for your attention. I realize that it's 4:01, and I need to take a flight. At what time is my flight? 5 o'clock? 5 o'clock. So guys, I'm sorry I have to leave.